My name is Karen Mongol. I'm the Bioinformatics Coordinator for the Analysis Groups here at the Genome Sciences Centre. One of the things that the Analysis Group is involved in is detecting structural variants, and so I'm going to talk a little bit more broadly about some of the things that are involved in that process. So my talk overview, I'm going to have three parts. In the first part, I'm going to give a bit of background. What is a gene fusion? How do they arise? And why are they important, specifically in cancer? In part two, I'm going to talk a little bit about considerations for bioinformatics tool selection for structural variant detection and what you can do when you have actually generated your data. And in part three, I'm going to talk a little bit about comprehensive structural variant detection and a tool that we've written to aid with that. Okay, so part one, gene fusions. What, how, and why? So I'm going to start with describing what a gene fusion is. A gene fusion is when two separate genes form together to produce a chimeric gene. The resulting product may lead to abnormal expression and gene function and can cause abnormal proliferation of cells and cancer development. So in the schematic, you can see that a breakpoint in the DNA is usually intronic and forms together to give a fusion gene. And at the RNA level, we would get what's called a fusion transcript, where exons generally match at exon-exon boundaries. What is a gene we've described, and how do they arise? So there are five very general ways in which gene fusion can arise. The first three involve just a single gene. The first of those is what we call a deletion event. And this is basically where part of the gene is actually deleted and lost. And assuming a diploid cell, we would expect to see a copy number change, or a copy number loss, in fact, for this type of event. For a duplication event, this is where a piece of the chromosome is actually duplicated and reinserted into the chromosome. In the schematic, we're showing what's called a tandem duplication, but in fact, the inserted sequence could actually flip and become an inverted tandem duplication as well. The third type of change is an inversion event, and again, this is on a single chromosome and involves a piece of the chromosome flipping and reinserting back into the same chromosome. This would be seen as a copy number neutral event because no DNA is lost or gained. The final two structural changes are an insertion event, which involves two separate chromosomes. So in the schematic, I'm showing an insertion of chromosome 4 into chromosome 20. Again, there is no DNA loss, and so there is no copy number change. And in the last case, I'm showing a translocation event where a piece of chromosome 4 translocates onto a piece of chromosome 20, and a piece of chromosome 20 translocates onto a piece of chromosome 4. In this case, it's a balanced translocation, and again, there is no DNA loss. So these are the five main rearrangement types that can arise and cause gene fusions. A little bit of history. So the first gene fusion was identified back in around 1960 and is called the Philadelphia chromosome. It arises when the 5' part of the BCR gene on chromosome 22 fuses to the 3' part of the ABL1 gene on chromosome 9. It was first discovered in chronic myelogenous leukemia and occurs in approximately 95% of CML patients. It exerts its oncogenic phenotype by constitutively activating the ABL1 kinase domain. In the schematic, you can see that the derivative chromosome 22 is actually called the Philadelphia chromosome and contains the BCR-ABL gene. So, gene fusions have been around for a little while. We've known about them from about 1960. There was slow progress in detection over the next few decades. The method for detecting these was cytogenetics, and is quite a low resolution detection method. But as we move towards the start of this century, we had the completion of the Human Genome Project, and we started to see that drug companies were able to design drugs to specifically target fusion proteins. And in 2001, we had imatinib, a tyrosine kinase inhibitor, which specifically targeted the BCR-ABL gene. In 2006 was the launch of the Cancer Genome Atlas project, which was a big effort to sequence approximately 500 cases of 20 different cancer types, and is a huge resource for the community now. And in 2008, there was a big change in the technology that we use for detecting genes, and that is next-generation sequencing. And since then, we have seen a huge increase in the number of gene fusions that have been identified across many different cancer types. So, what do we know about gene fusions from the data that we have currently? So, one of the things that we have observed with gene fusions is their recurrency, and specifically in different cancer types. So, for the FGFR3 TAC3 gene fusion can be seen in glioblastoma, it's seen again in lung cancer, and also in bladder cancer. But not only do we see gene pairings that reoccur across different cancer types, we see the same gene with different partners in different cancer types as well. So, for example, FGFR3 is partnering with AFF3 in breast cancer, and as well with BAI2PL1 in bladder cancer. So specific genes can partner with the same gene or with different genes. So just to summarize some of the things that have been observed, we can look at fusion functionality. So we know that gene fusion landscapes are quite diverse. So gene fusions vary across different cancers and across individuals with the same cancer. You don't see the same fusion necessarily in all the individuals with the same cancer. The pairings that we see can be quite specific. The genes tend to fuse with very few partners, and different cancer types and different fusion networks have different fusion networks. The frequency of gene fusions varies considerably with different cancer types. In general, gene fusions are quite rare, but can be the dominant oncogenic driver, and often anti-correlate with other somatic mutations known for that cancer type. Gene fusions tend to have specific functions, for example, activating the kinase domain or the binding it domain in their fusion gene. So looking a little bit at the fusion proteins that are generated. So the breakpoints for the protein tend to be in disordered regions, but maintain the reading frame and the protein structure. Fusion proteins are relatively depleted in domains, so the proteins that form fusions have relatively few domains compared with the fusion transcripts, which encode more domains than you would expect by chance. Fusion protein architecture is non-random and can lead to novel architectures, and disorder is seen in fusion proteins, and this could promote viable joining with different domains with a view to oncogenic driving of the cancer. So why are gene fusions important in cancer, and why is it important to detect them? So the first reason is prognostic significance. So for example, the TMPRSS2 ERG gene fusion in prostate cancer, when associated with prostate-specific antigen levels, can be an indicator for recurrence and shortened time to recurrence. So it's particularly important to identify these patients because they are much more likely to relapse and relapse quickly. So by identifying this gene fusion, we can identify the specific patients that are at higher risk of prostate cancer relapse. Diagnostic significance is also important. It can give you confirmation of diagnosis. As I said previously, the BCR-ABL gene fusion found in CML patients is the hallmark fusion for this cancer, and is expected to be seen in approximately 95% of patients, and this confirms the CML diagnosis. In another case, these gene fusions can be specific to a subtype. So the EML4 ALK fusion, for example, is seen in a subgroup of non-small cell lung cancer, and that's about 5% of patients. And it's very important to identify these 5% of patients because this has therapeutic significance. So this gene fusion arises in lung cancer and is from an inversion event on the P arm of chromosome 2. It is usually found in patients that are non-smokers or very light smokers, and often occurs in younger patients. The reason it's important to identify patients with this rearrangement is because these patients do not benefit from the current standard drug therapy, which is tyrosine kinase inhibitor therapy. The ALK fusion means that they are much likely to respond to an ALK inhibitor, for example, crizotinib. And if this ALK gene fusion is identified using an FDA-approved test, these patients would be given an ALK inhibitor instead of standard therapy. The presence of this ALK gene is also mutually exclusive with other mutations that are normally found in lung cancer. And the methods for detecting the ALK rearrangement are often run in conjunction with tests for EGFR and KRAS mutation, just to molecularly identify everything that could possibly be driving the cancer in these particular patients. So as I said, it's very important to identify this particular gene fusion because these patients would be given crizotinib and are resistant to standard therapy. So in part 2, I'm going to look at bioinformatic tool selection and what you might be able to do with the results once you've identified some gene fusions. So there are various ways to detect gene fusions. A lower resolution way is fluorescent in situ hybridization. So in this method, a red and green probe are hybridized to the ALK gene. This is looking specifically at ALK rearrangements. And when you look at the individual cells, you are hoping to see, as in the bottom part of the picture, cells that have separated to give a separate red and green fluorescence. This is called break apart and indicates that the ALK gene has been rearranged. And this would be identified as a positive rearrangement and is part of the clinically FDA-approved test for ALK rearrangements. It's also possible to detect gene fusions using polymerase chain reaction. In this case, sequences are designed based around the breakpoint for the gene fusion. and the resulting PCR DNA is amplified, and then, for example, sang is sequenced to give the breakpoint position. This can be run across many different samples, but assumes that the breakpoint is consistent across samples. More recently, with the next generation sequencing, we have moved to massively parallel sequencing. And in this example, I'm showing Illumina-based sequencing, which is sequencing by synthesis. In summary, what happens is the genomic DNA is shared. Adapters to the DNA makes the sequencing library, which is applied to a flow cell. Cluster generation by bridge amplification is done actually on the flow cell. And these flow cells are loaded onto the machine, and then sequencing by synthesis is performed to produce the individual reads for analysis. So what we generally do at the GSC is Illumina sequencing. So the rest of the presentation is based on detection of gene fusions from Illumina sequence. The first way that we detect gene fusions is through a targeted fusion detection. So in this case, what we do is create a 50 base pair probe that contains the event of interest. This event can be an SNV, an indel, or even a fusion breakpoint. These probes are created, and then using the target FASTQ or BAM file, we screen for presence of these particular mutations in the patient library. The advantages of this is that it's very specific to the events that you're interested in, and it's very fast. This can complete and give you results in approximately 2 hours. The disadvantage of this is that you need to know ahead of time what you want to look for in the particular sample. Another method that we use is de novo fusion detection. And in this image, I'm showing de novo fusion detection by assembly. So we start with the individual paired end reads, which we assemble into a contig. That contig is then aligned to the reference genome, and the reads are aligned back to the contig. And from this, we can identify contigs that show chimeric alignment to the reference and individual reads that support the presence of that chimeric breakpoint. The advantages of assembly-based fusion detection are it's very comprehensive. You can identify all the different types of structural rearrangements that occur. It has high specificity because contigs themselves are assembled from the reads representing the event. And because it generates contigs, there is better interrogation of the breakpoint itself, which means you can look to see whether there's any mistemplated sequence at the breakpoint, which would give you information regarding the method by which the break had occurred. The disadvantages are that it requires a large resource requirement and is multiple step process and can be quite slow. And because contigs are generated, it is generally a lower sensitivity method. The third method that we utilize is de novo fusion detection by alignment. So in this case, the paired end sequencing reads are aligned to the human reference. We use BWMM as our aligner for genomes, and what the structural variant caller is looking for is mapped discordant reads for the SV prediction. It looks at the evidence for the prediction, and at the end of the day, it produces structural variant breakpoints. Some tools are able to do a local assembly, which will also give you a breakpoint contig, whereas other tools detect variants by, for example, flanking reads, so there is no specific breakpoint call itself. The advantage of this method is that it's fast with lower resource requirements and gives higher sensitivity. However, it does have lower specificity because you're making many more calls and you have to get your filters correct to get through the gene fusions you're interested in and remove the lower evidence gene fusions. So some considerations for tool selection are dependent on the project that you're looking for. So for example, for sample source, are you looking at fresh biopsies or are you looking at archival samples? Important to know this because, for example, with archival samples, the fragment size tends to be much shorter, and depending on the read length of the sequences that you're using, this can affect the ability of the tool to detect structural variants because it has to take into account that reads could possibly overlap with shorter fragment lengths. Data type is also important to know. Are you working with DNA or RNA? In general, tools are specific to either DNA or RNA. They don't tend to work on both. So you need to select your tool dependent on your data type. You also need to look at what your input is going to be. Are you going to be using a FASTQ or a BAM file? And again, read length is important. Some of the early tools do not work well with longer read lengths that we're now seeing of, for example, the latest versions of Illumina sequencing machines. For test samples, it's important if, for example, you're looking at match samples. A lot of the projects we do here at the Genome Sciences Centre are patients with matched DNA, tumour and normal samples as well as RNA samples. And what we want to do is leverage the information from all our samples to look to see which of the gene fusions are actually expressed in that particular patient. We also do work with rare diseases, and we could be looking at individual analysis, for example, using trios of data. So in these cases, we would get a proband where we're interested in finding what's unique to the proband by screening against both the mother and the father. So test samples are important to look at as well. So when we're actually looking at the tool itself, some of the considerations that we have are sensitivity of the tool. How sensitive is the tool to events that we might be seeing? Are we looking at low tumor content samples? In which case, we might want to choose tools that we have tested and can say that they are more sensitive. We tend to run our tools through a test data set. So we use Colo 829, and we can compare sensitivity and specificity of individual tools based on known events in Colo 829. We're also interested in the speed of each of the tools. Now, tools can be very sensitive, but how quickly do they complete the task? This will depend on the resources that you have available. Are you going to be running your tool on your desktop, or are you going to be running it on a cluster? These are important considerations when selecting your tool. Another important consideration is how deterministic is the tool. You would hope that running the same tool with the same input on different days would give you exactly the same answer. But for example, we know that assemblies are not quite deterministic. They are a bit stochastic in their output. So we could find that if we changed our assembly input, that we might get slightly different analysis results. But knowing this helps us with our analysis. So once we have the data, what do we want to do with it? one of the things that we like to do is visualize the data. So in this slide, I'm showing circles, and this is a very useful tool to give you an idea of just how rearranged the genome is for an individual patient. So in this case, in the middle, we're seeing all the translocation events in the genome for this particular patient. And moving out from the middle, we can see each of the individual inversions, insertions, and deletions relative to the chromosomes, which are the colored bands further out on the circle. We have the matching transcriptome for this particular individual, and we can see from this which of the rearrangements in the genome are expressed in the transcriptome. And as you can see, there's much less going on in the transcriptome. Another visualization that we can do is the visualization of the gene fusion itself. In this slide, we're showing breakpoints for the gene fusion at the chromosome level at the top. The next part down, we're showing the breakpoints relative to the genes themselves. And then we can see the breakpoints relative to the transcripts of each of those genes and each of the domains that are associated with those genes. And finally, at the bottom of the screen, we can see the predicted fusion transcript that is generated from gene fusion, and we show the domains and the reading frame for that gene transcript. So another thing that we can do with the genes once we've identified them is to look at existing databases to see, has this gene fusion been seen before, and if so, what cancer datasets has it been seen in. One of the recent publications is from the Jackson Lab on the Tumor Fusion Gene Data Portal. In this data portal, they have reanalyzed all the TCGA datasets using Prada and have put them into a database, which is a very nice interface where you can look specifically for your gene of interest, you can look at your fusion of interest, you can look by TCGA sample. It's a really nice interface to do some digging further into your gene fusion of interest. Another useful resource is COSMIC, which is hosted at the Wellcome Trust Sanger Institute. COSMIC is a curated resource where they have not only gene fusions, but they have somatic variants, and they have also a lot of information about cell lines as well. So it's useful to see whether your gene fusion has been seen in a cell line and could lead to further functional work using a cell line. Another database of interest is the Mitelman database of chromosome aberration and gene fusions. This combines cytogenetic information with SNP arrays together with curated information on gene fusions. Again, it gives you a different set of results to look at and is another useful resource for looking for your gene fusion of interest. So moving on to part 3, this is going to summarize some of the conclusions that we've made having analyzed thousands of samples for structural variant detection. Comprehensive structural variant detection for us involves multiple tool input. We've found that we can improve our sensitivity and specificity by using many different tools as input for our structural variant detection. This leverages their advantages and enables us to look more specifically and with better sensitivity at rearrangements in a sample. But what we need to do, because we have so many tools as input is cluster the breakpoints that are called. Sadly, tools do not call the exact same breakpoint, even though they may be calling the same event. And so you need to cluster the breakpoints in order to prevent overcalling of events in your sample. What we want is a consistent breakpoint calling method. As I said, different tools call their breakpoints in different ways. Some use contigs to call breakpoints, and others use flanking or split reads. In any case, what is ideal for us is a consistent method for calling a particular breakpoint. As I said previously, we do a lot of DNA and RNA analysis, and so what we want to do is pair that data together to give a better overall picture of what is going on at a structural variant event in a particular patient. And we want to look at the evidence that supports these events. Different tools use evidence in different ways. We want a standard way to look at the evidence and know that we can adjust our filtering based on the evidence support for particular events. We want to be able to use a standard annotation. Different tools use different annotations, and this makes it more complicated when trying to report gene fusions. So by using a standard annotation method, we can report with confidence the gene fusions that are found. And finally, what we want is a standard output format. A lot of these tools have many columns in their output format. A lot of these are not necessarily useful for an analyst when looking at the data, and it can make it very difficult to parse that data out to do, for example, a cross-cohort analysis. So what we have come up with at the Genome Sciences Centre is our own tool called MAVIS, which merges, annotates, validates, and illustrates structural variants. This slide shows the MAVIS process outline. So there are 4 main modules to this process. In the first module, we look to cluster the breakpoints from the input tools. We then filter those based on the proximity to annotations and call a new merged breakpoint pair from the group. We then gather the read evidence in the validate module and call the breakpoint pairs. These breakpoint pairs can be called based on a contig that is generated on split reads or on flanking pairs. We then annotate with gene and transcript level information and build fusion transcripts for exact calls. These are then drawn with SVGs and can be later accessed. The pair calls are made between the libraries. So, for example, with a DNA-RNA pair, we would be interested in somatic events, we'd be interested in germline events, and events that are expressed in the RNA. And then finally, we have a standard output val with HGVS nomenclature, so we have no confusion about the genes that are being called in the gene fusion. To look in a little bit more detail at these separate modules, we start with merging. So in merging, we have a converter script that takes the input from any SV caller, as long as it is put into a common format, which this script can do. It needs very basic information from the SV caller, and that is then used. What we do next is filter based on user-defined masked regions. So, for example, these regions could be centromeric regions. These are standard tracks from the UCSC browser and can be used if the user wants. These don't have to be used. We then split the call by type. We want to separate the different types of events. So we would have deletions and translocations, etc. And this is important because we then merge our events based on proximity. But But we wouldn't want to merge deletions and translocations with breakpoints in the same location because these are different events. We merge based on a click finding algorithm and follow this with hierarchical clustering. For the validation module, we use the VAM file to collect support for the input event calls. This uses repair fragment distribution to define the intervals where reads will be collected from. This is important because, as I said previously, if we're using FFPE data, we would expect that interval to be different to a fresh frozen sample that has a larger fragment distribution. We collect spanning reads, split reads, and half-map reads, as well as flanking and compatible flanking pairs for our evidence gathering, and we standardize the SCAR and read alignments to ensure that we have reproducibility in our calls. We also use a collapsed annotation model so that we can calculate repair fragment sizes for transcriptomes. We then perform a local assembly to generate contigs, and then start the calling by breakpoint pairs. We call in the first instance by contig. If a contig has been assembled, then we call by split reads, and then we call by flanking or by flanking and split reads. By doing this, we can increase our sensitivity of calls that are made. We follow this with an annotation module. We look at the gene level, and in this we look at the nearby genes to the breakpoint. We look at the genes encompassed by the event. For example, a structural variant event could occur in intergenic regions, but may still be important if it's deleting, for example, oncogenic genes. We also look at the genes at the breakpoint. At the fusion transcript level, we look at the exon and intron of the breakpoint. We use a splicing model to predict if the fusion will be in frame or out of frame. And we also predict the domains that are retained by realigning the protein domain sequence to the new amino acid sequence of the fusion protein. We follow this by an illustration. So for any fusion with a breakpoint level resolution and putative splicing products formed, we can produce a standard figure. Again, we show the chromosome level, the individual gene level, and the transgate level, and finish that with the actual fusion product. This is a nice standard way to generate a figure for your gene fusion of interest. We can also model alternative splicing. So in this diagram, we have a G2A splice site mutation seen in the genome, which is linked to an exon 9 skipping event in the RNA, and we've overlaid the expression level analysis to show that the expression for exon 9 is lost, which ties in with the splice site mutation and the exon skipping event. So these can all be tied together in a nice diagram. So this is a tool that we've created, and if you want more information on this, it is open source. The tool is MAVIS. It is available on GitHub. We have a user support group who is able to answer any questions that you may have that is not part of the documentation, or any issues you may have installing the software. We have recently submitted this tool to Bioinformatics, and we are presenting this at AGBT 2018. So this is definitely available for anybody who is interested in looking at this. So with that, I come to my acknowledgements. None of this is possible without the work of many teams here at the Genome Sciences Centre. And so the acknowledgements marks the end of my presentation.